Hello everyone, welcome to my YouTube channel Microbiology Simplified. I am Dr. Amit Kr Singh, MD Microbiology. This is the second video on my pathology lecture series. So the first video was on the morphological form of the parasite, that is the cyst. Now this is the second lecture on the morphological form of parasite, that is the trophozoite. So as I told you, the cyst form is present in those protozoan parasites which can survive in external environment, but this is not in the case of trophozoites, because trophozoite can be seen in other protozoan parasites too. So what are the protozoan parasites in which we can see the trophozoite forms? So trophozoite are present in all protozoan parasites except hemoflagellates. So amebs, and that is intestinal free living, lumen dwelling flagellates, sporozoas, that is hematozoa and coccidia, and ciliate, all of them contain the trophozoite as their morphological form. But hemoflagellate, that is leishmania and trypanosoma, do not have trophozoite forms. Okay. So leishmania and trypanosoma, both of them do not have trophozoite form, the life cycle. Okay. So let's see one by one the trophozoite of various protozoan parasite, how they will look like. I will tell you the characteristic features and how to identify them. Okay. So let's see. So the first image, if you see the shape is, I will just write the characteristic and then I will tell the name of the parasite. The shape is amoeboid, irregular shape. There is no regular shape, not circular or not any characteristic pear shape or tennis racket shape. It is amoeboid shape, irregular shape. Okay. And then what else you can see? You can see the number of nucleus. The number of nucleus in this case, it is single. So it is uninucleate. Okay. And they have got this pseudopodia like projection. This pseudopodia is basically their organ of locomotion, locomotory organ, and by virtue of this pseudopodia they have got characteristic amoeboid motility called as gliding or crawling motility. Okay, gliding or crawling motility. So all trophozoite in which there is no flagella, those trophozoite will have gliding motility by virtue of pseudopodia. Remember this. What else we can see? Then see for this, the presence of this. What is this? These are ingested RBC. So the trophozoite of Entamoeba histolytica ingest RBC, and ingested RBC can be seen inside them. This phenomenon is called as erythrophagocytosis. So this is characteristic of trophozoite of Entamoeba histolytica. So it is amoeboid in shape, uninucleate, shows gliding motility and shows erythrophagocytosis. Okay, so whenever you will see a trophozoite in which we can see RBCs, so and also the shape and nucleus, if all the thing matches, then it indicates the trophozoite of Entamoeba histolytica. Okay, now coming to the next image. In the next image, if you see, there are two forms. The first form, this one, and the second form, this one. The first form looks like similar to the trophozoite of Entomobacterica. This is the amoeboid stage of this trophozoite. And the second form, if you see, there are flagella present. So this is the flagellar stage. So the only amoeba which has got two forms of trophozoite is Naegleria fowleri. Okay, Naegleria fowleri trophozoite has got two forms, amoeboid forms and flagellar form. Out of this, amoeboid form is the infective form. Okay, infective form is amoeboid, whereas this flagellar forms helps in spread of Naegleria fowleri to new water bodies, and once it will reach to a new water body, so it can again transform into amoeboid form. So this flagellar form can transform into amoeboid form, and that's why this Naegleria fowleri is also called as amoeboflagellate. So if you remember, I told you another amoeboflagellate, that is Dientamoeba fragilis. Okay. So this is the characteristic of trophozoite of Naegleria fowleri. Fine. So Naegleria fowleri has got two forms of trophozoite, amoeboid form and flagellar forms. Flagellar form is biflagellates. You can see the two flagella. It is biflagellate, whereas there is no flagella in amoeboid form, and the motility in amoeboid form is by virtue of pseudopodias, and this pseudopodias is basically round in shape, and that's why they are called as lobopodia. Okay, so this is about the trophozoite form of Naegleria fowleri. Now coming to the next image. So if in this image, if you see, there is something spine-like projection. This is spine-like projection which is seen in this structure. This is called as spine-like pseudopodia, and also called as acanthopodia. Acantho means spine-like, and that's why the parasite is called as acanthamoeba. Okay, and it is also uninucleate. So all the trophozoites of amoeba, either intestinal amoeba or free living amoeba, the number of nucleus is single. Okay, the only difference is in the presence of flagella in case of Naegleria fowleri and presence of spine in case of acanthamoeba. So now coming to the next image, this shape is not like amoeboid. This is typical shape is like tennis racket shape, and this, if you see, this trophozoite is, if you divide it in two half, it is bilaterally symmetrical. Okay, so because of this bilateral symmetry, it has got everything in pairs. So it has got one pair of nucleus, that is total of two nucleus, but as it is bilateral symmetrical, so we call it one pair of nucleus. Okay, and four pairs of flagella, so total two nucleus and total 8 flagella, but in pair. Okay, and the motility which is seen in case of this is falling leaf. So what is this? This is a trophozoite of Giardia duodenalis. This Giardia duodenalis is also known as Giardia lamblia and Giardia intestinalis. Okay, Giardia lamblia and Giardia intestinalis. So, what are the features? Tennis racket shape, also known as pyriform shape or heart shape. Okay, and it has got one pair of nucleus or two nucleus and four pairs of flagella, that is 8 flagella. So where are these flagella located? One is anterior, one is posterolateral, one is ventral, the last pair is caudal. So if you can see, I'll show all the four pairs. So this is anterior, this is posterior, this is caudal, ventral, and these two are caudal. Okay, so 1, 2, 3, 4, four pairs of flagella. And what is the type of motility? Falling leaf motility. How to remember this? So a small trick you can use. Giardia sounds like garden, and in garden we can see leaf. So falling leaf motility in case of Giardia. So if you want to remember this, you can remember it like this. Okay, fine. So now let's move into the next image. So now this looks somewhat similar to that of Giardia, but the shape in this case now is not tennis racket, it is either pear shape or oval shape. Pear shape or oval shape. So I will just draw it. And it has got single nucleus. And apart from this nucleus, it has got four anterior flagella. 1, 2, 3, and 4. And a fifth flagella which actually runs along the outer margin of undulating membrane. So this is the undulating membrane, and the fifth flagella runs along the outer membrane of outer margin of undulating membrane. Undulating membrane is basically a cytoplasmic extension of the cell itself and helps in movement. So this is undulating membrane, and these are the four anterior flagella and one flagella along the undulating membrane. So they have got total 5 flagella. Okay, what else? Apart from flagella, they have got an exostyle which runs throughout the body and exits posteriorly like a tail. So this is the exostyle. So it has got a pear shape, 4 anterior flagella, 1 flagella which runs along the undulating membrane, so total of 5 flagella, and exostyle which comes out posteriorly like a tail. And it has got a characteristic motility. Okay, and this motility is known as twitching motility or jerky motility. So to remember that Trichomonas vaginalis has got twitching motility. So just see, twitching starts with T, Trichomonas starts with T. So these two matches to each other. So Trichomonas vaginalis has got twitching motility or a jerking motility. So remember these points: pear shape, 4 anterior flagella, 1 flagella running along the undulating membrane, and an exostyle and twitching motility. Fine. Now the next image, there are two images you can see, first, second. So these two images are the trophozoite of Plasmodium, and the first image is the early trophozoite form, early trophozoite form, early, and this is late trophozoite form. Okay, so you can see in early trophozoite forms the trophozoites look like a ring. Trophozoite looks like ring. So, and this is present inside RBC. This is RBC, and this trophozoite is present inside the RBC. And how it will look like, I'll draw the diagram. So if this is the RBC in which the trophozoites are present, so this, it will look like a ring. So this is the stone of the ring and this is the margin of the ring. So basically it occurs because of presence of vacuole in the cytoplasm, and this vacuole pushes the nucleus to the periphery and the cytoplasm make a structure like ring. So that's why the ring, it is called as the ring form, or the early trophozoite form appears like a ring. And this ring form differs from species to species. So just few differences between the species of Vivax and Falciparum, I tell you. In the ring form, first, in Vivax the size of the ring is approximately 1/3 to half of the size of RBC, whereas in case of Falciparum it is about 1/5 of the size of the RBC. So that means the ring form is smaller in case of Plasmodium falciparum. Also, the number of rings in case of Vivax is usually single, whereas in case of Falciparum it is mostly multiple. Thus, the size of RBC, the infected RBC, is usually enlarged in case of Vivax, whereas in case of Falciparum it is usually normal in size, the RBC, the infected RBC. Okay. And also we can see in the RBC of Vivax dot structure which, because of alteration of RBC, those are called as scuffness dot in case of Vivax and Maurer cleft in case of Falciparum. Okay, so this is the difference between the ring form or early trophozoite form of Vivax and Falciparum. Now the second structure, if you see, this looks more amoeboid in shape. So this is the late trophozoite stage seen in Plasmodium, and it is more amoeboid in case of Vivax and more look like ring form, a compact or more denser form of ring form in case of Falciparum. And you can see the scuffner dots very well in this image. The late trophozoite is amoeboid stage of Plasmodium Vivax. Okay, so these are the trophozoite stage of Plasmodium. Fine. So now move into the next trophozoite, and this is the trophozoite of the coccidian parasite, that is Toxoplasma gondii. Toxoplasma gondii. So the characteristic shape of this trophozoite is crescent shape, and this trophozoite, as it is rapidly multiplying trophozoite, so they are called as tachyzoites. Okay, which parasite parasitic form has got a crescent shape? So the gametocyte of Plasmodium falciparum, this has got a crescent shape or banana shape. Okay, crescent shape or banana shape. And this stachyzoid, if you draw the diagram, I told you crescent shape. So it has got a single nucleus, single large nucleus, single large nucleus, and tapered anteriorly and blunt posteriorly. Okay, so this is the characteristic trophozoite of Toxoplasma gondii, which is known as stachyzoid, as they are rapidly multiplying. So what is the characteristic shape? Crescent shape. Okay, now coming to the next image. This is the largest protozoan parasite, and if you remember in the last lecture I told you the largest protozoan parasite is ciliate. What is the name of the ciliate? Balantidium coli. Balantidium coli. Okay, and this trophozoite has got certain characteristic which I can start with the letter B. So the name, it starts with B, Balantidium coli. Now see what are the feature which starts with B. First, as I told you, it is the biggest trophozoite, around 60 micron in diameter. Then it has got two nucleus. It is binucleate. One macronucleus and one micronucleus. And this macronucleus, this one, this is bean shape. Okay, this macronucleus is bean shape. If you see in this diagram, the bean shaped macronucleus can be seen, and also you can see an opening there. This opening, which is present in case of trophozoite of Balantidium coli, is called as cytostome, opening of the cell, cytostome. Okay, so these are the characteristic of trophozoite of Balantidium coli. B, Balantidium coli, B is the biggest, B is binucleate, and the bigger nucleus is bean shaped, and it has got a characteristic motility which is called as spiraling motility. Spiraling motility is seen in Balantidium coli. So just a quick recap of whatever I taught you in this video. So first of all, let's see the motility. In Giardia we saw falling leaf motility, in Trichomonas vaginalis we saw twitching motility, and Balantidium coli we saw spiraling motility. Now, which of the trophozoite are uninucleate? Amoebs uninucleate, either intestinal or this free living. Trichomonas uninucleate, but not the Giardia. Okay, then in case of uh, this Plasmodium, again it is uninucleate. This Toxoplasma uninucleate, but Balantidium coli is binucleate. So binucleate is Balantidium coli and Giardia. Okay, they have got two nucleus. In Giardia it is pair of nucleus because of the bilateral symmetry, and in case of Balantidium there are two nucleus, but one is big and one is small. The bigger one is macronucleus, which is bean shaped. Okay, and the characteristic shape which is seen in case of Toxoplasma gondii, it is crescent shape, whereas the characteristic shape of the trophozoite of Giardia, it is tennis racket shape. Of Trichomonas it is pear shape, and in case of Acanthamoeba it is spine like pseudopodia, but there is no characteristic shape because it is amoeb. And what else? In case of Plasmodium it is ring shape, signet ring shape, because of the vacuole which push the nucleus and cytoplasm to the periphery. Okay. So I hope in this video you would have understand the various morphological forms of the trophozoites of the protozoan parasite, and you will be able to differentiate on the basis of the image if it is given in the examination, or on the basis of the characteristic feature mentioned in the question. So I hope if you'll follow this video lecture series, so you'll be able to understand the basic concept of the parasitology, and will be able to answer the questions asked in the examination, either image based or question based on the characteristic feature of this morphological forms and the disease caused by the parasites. So stay with these videos, and please go through all the videos, then only you'll be able to get a better concept of the parasites, especially the protozoan parasites. So I hope I was able to explain the topic well to you. So if you like the video, stay with the video, follow me, and I wish all the best to you for your examination. Take care. Bye bye.